Hi, my name is Terry Sanger. I'm a child neurologist and I work at Children's Hospital of Los Angeles. I want to talk about deep brain stimulation. Deep brain stimulation, or DBS, is a treatment that can be used for a variety of brain disorders. It's most commonly used in adults with Parkinson's disease, but it can also be used for children with certain forms of dystonia. Dystonia is a symptom that can occur in many types of brain injury, but particularly with brain injury to a deep area of the brain called the basal ganglia. And when children have dystonia, they try to move and they get the wrong combination of muscles. So instead of activating one muscle, maybe 6 or 7 muscles activate simultaneously. And as a result, the arms or legs will twist, or the trunk or the neck will twist in ways that they don't want. Anytime they try to move, they can relax, but the attempts at movement always lead to a distorted posture or a distorted movement, which can involve shaking or twisting movements, and when severe, this can essentially prevent any type of functional movement. It's unfortunate because there are not a lot of good treatments for it. Now, it turns out that deep brain stimulation has the ability to almost completely fix certain types of dystonia, particularly ones that are due to genetic disorders. In cerebral palsy, the dystonia is due to injury to the brain, and usually there's not injury just to a single area of the brain, but to multiple areas. So even if we can fix the dystonia, usually there are other movement problems, including, for instance, spasticity or weakness, or other types of problems, ataxia, dyspraxia, bradykinesia, which is slow movement. And those will not really respond very well to deep brain stimulation, or at least not in the same areas that will respond to dystonia. Now, the deep brain stimulation technique involves putting a wire into the part of the brain where we think the signals are coming from. And the signals, we think that there's too much activity in some part of the brain because there's too much muscle activity that has to be generated at some point by too much neural activity in neurons within the brain. And the idea is that we can somehow stop that or block it in a way that prevents the bad activity from getting out. The brain is not working as it should be. It is generating too much activity, but if we can block the chain that connects the abnormal neural firing to the abnormal muscle firing, then perhaps the muscles will relax and improve. And so the idea is to put wires into those areas of the brain. The wires are tunneled under the skin to a pacemaker in the chest, and then from that pacemaker in the chest we can generate signals, very regular pulses, for instance short pulses occurring 100 to 200 times per second. These electrical signals go into the brain. They depolarize neurons, which means that they make the neurons fire. They force them to fire, actually in a very abnormal pattern, because it will force a large group of neurons to all fire at exactly the same time. That's very unusual. But for whatever reason, this does seem to be able to improve dystonia in children. It doesn't work in everybody, and the different targets may be needed in different children because there are many possible brain regions that can cause dystonia, just as many brain regions can lead to movement and can cause muscle contractions. Abnormal activity in many different brain regions can cause abnormal muscle contractions. And so the tricky part is to find exactly the right place to put the wires. Now, the thing that is most surprising about this is brains, of course, are very complicated devices. There are on the order of 100 billion neurons, trillions of connections between these neurons, and the information flow is huge. Every single child's brain has the ability to process more information than the entire internet, and it's doing this constantly all the time. So how could we imagine that a battery connected to essentially pacemaker technology, the same technology used to control your heart, that is just putting out a regular pulse, how could that have any type of beneficial effect on changing the patterns of activity? We don't know the right patterns of activity. I can't put in a thought. I can't put in a skill or a muscle pattern. All I can do is use this to modulate brain activity. And that's the key. It's called neuromodulation. And the idea of neuromodulation is that we modulate, we change the ability of these areas of brain to fire without necessarily telling them exactly what to do. And it's a very gross type of intervention, but if we think about it, most of our interventions are like that. If I give a medicine that might control dystonia might include something like diazepam or baclofen or trihexyphenidyl. If I give these medicines and you take them by mouth, they go into the child's blood, it circulates to the brain and affects every single neuron in the brain that is able to respond to those chemicals. So it's a very broad, very non-specific type of effect of these medicines. So all the medicines do is modulate. What deep brain stimulation does is it modulates very locally. It's near the tip of the wire, near where the electrode is, it modulates, and it will modulate all the neurons, or many of the neurons, in that area. Whereas a chemical, trihexyphenidyl, for instance, is something that blocks acetylcholine, one of the neurotransmitters used in the brain. Acetylcholine is not used by every neuron, so that trihexyphenidyl will only block the acetylcholine using neurons. So the drugs tend to be chemical specific or transmitter specific, or in some cases cell type specific, whereas deep brain stimulation is a modulation that is location specific, because we put it into a very specific location, but within that location it has the ability to stimulate many different types of cells, and could potentially stimulate cells that use acetylcholine as well as cells that use GABA, which is an inhibitory transmitter, or cells that use glutamate, which is an excitatory transmitter, or any of the other cells that are in that area. But no matter what we do, either with chemicals or with deep brain stimulation, this is a modulation. So why is it possible that modulation could improve or reverse the effects of a disorder, such as cerebral palsy or a metabolic disorder or other types of genetic brain disorders? Well, the reason is that the disease itself is a modulation. Diseases don't inject information either. They don't care about the specific skills that they are impairing. They don't even care whether a particular neuron that is being injured affects the arm or the leg or speech. All they do is injure neurons, and they can injure things in a very location-specific way, which happens, for instance, in a stroke. If there's a blocked blood vessel, the neurons in the region of that blood vessel will die, and everything in the region of that blood vessel may die. Or if you have a genetic disorder that affects a certain type of neuron, every neuron of that type might die or malfunction in some way. And there can be developmental disorders where whole regions of brain or whole types of cells don't get to where they're going to go or do not develop normally. So the diseases have, in some ways, a very similar kind of profile to the way in which we have these interventions. And this is fortunate because it means that, in fact, these interventions, which essentially are not just neuromodulation, but are almost could be thought of as demodulating the disease itself is a modulation of brain function. At the most extreme, a disease modulates brain function by preventing it, by completely blocking it. For instance, a stroke could kill every cell in a region, and therefore no information flows through that. If we then put an electrode in that area and try to modulate it using deep brain stimulation, it's not going to help. There's nothing to stimulate. But it may be that the effect of that stroke on that region of cells is such that the areas that those cells would have connected to are now being modulated improperly. So if, for instance, the stroke were to cause death of inhibitory cells, then everything that they would have connected to is now disinhibited and may fire too much. And so we may be able to use the deep brain stimulation or a chemical or some combination to unmodulate that, to again lower the firing, lower the activity of this group of released cells, of cells that are now disinhibited. And the opposite can happen as well. If a group of cells is killed that normally would have excited other cells, then we may be able to use the deep brain stimulation to excite those cells, essentially to make it more likely for them to fire so that the regular signals can come through. In both cases, but the disease and the treatment are not injecting or changing the specifics of the information that's going through. They're just changing the ability of the information to go through and the way in which it's processed. Eventually, of course, that does lead to differences in the pattern of information in the brain, and those patterns are expressed in abnormal muscle patterns and hopefully in partial correction of those abnormal muscle patterns by our interventions. The overall benefit of this, of course, is that in some sense the treatment matches the nature of disease. And this can be true whether it's a genetic disorder or whether it's an acquired disorder. And in some cases, there may be a global abnormality of cellular function, for instance, maybe a genetic abnormality that affects many cells, could potentially be treated by deep brain stimulation in a local area, and vice versa. A local injury could potentially be treated by a global use of a medication. When we think about this, the way the disease happens, whether the disease affects one cell type or just one region, the effects of that will propagate throughout the brain. They'll propagate because the abnormal pattern of signals in one area of brain, the rest of the brain was depending on those signals. Not every part of the brain. There might be a sensory part of the brain that is unaffected by abnormalities in the motor part of the brain. But wherever the abnormality is, will propagate. Eventually, of course, it has to propagate to muscles. Something that doesn't affect muscles in any way has no observable effect on behavior. That's more of a theoretical question because, of course, we care about thought and we care about feelings and sensation. But from the point of view of those of us who are interacting with the child, those thoughts and sensations eventually need to be expressed as some abnormality in muscle or behavior or response. Otherwise, we don't know they're happening. So at some level, we can say these patterns eventually have to make their way out to muscles. And our goal is to either block those patterns or somehow convert them into patterns that are less maladaptive, that are more functional for the child. Now, some patterns may be minor and we don't have to worry about them so much, and some patterns may be major, and sometimes we can only affect parts of the pattern, and so we will try to target something specifically. We might say, for a child whose arms and legs are affected, we might try to target their arms so that they can use a communication device better, and then use something else to look at their legs. We might put in more than one deep brain stimulation electrode to try to treat different types of symptoms. But the important way to think about this is that there's a pattern of activity which is distorted by the disease. The same thing is true for the intervention. When we put a deep brain stimulation electrode in somewhere, we are generating essentially a very abnormal pattern of neural activity. If I am stimulating a group of cells at 185 cycles per second, and I cause a large group of cells to depolarize that fast, that's extremely abnormal. The closest thing to that would actually be epilepsy, a seizure, where you're getting completely repetitive patterns. It's an information-free pattern. 185 cycles per second repeating over and over and over again, with all the cells in the area that are depolarizing at exactly the same time. That's a very boring pattern. There's no information there. And that may be exactly the point, which is that it may block or hide the information. It's as if you played a very, very loud and very, very boring song over a very interesting speech. You can't hear the speech. All you hear is the very boring music, and as a result, the information that was supposed to be there doesn't get through. When you substitute the boring information for the good information, now that boring information will propagate. The signals, the 185 hertz that we put in, will stimulate the cells near the electrode. Those cells, some of them will fire, and they will stimulate the cells that are further downstream from them. They may cause those cells to fire, or they may inhibit those cells, depending on whether those are excitatory or inhibitory neurons. Whatever they do, they're going to change the pattern of activity that occurs in the brain and the response of the brain to that. And so the idea is our neuromodulation, whether it's done by a medication or by deep brain stimulation, is trying to change one pattern in the brain, the pattern caused by the disease, into another pattern in the brain, which is the pattern caused by the disease plus the stimulation. And we hope that that second pattern leads to better muscle activity and better function than the first pattern. We need to be able to test this, and these are things that are very hard to predict, although eventually the hope is that we will get to the point where we can predict these. Another effect of these patterns, of course, is that the brain is always learning. And learning involves the learning of new skills that the child has to do during development. And a child with a movement disorder such as dystonia or spasticity or weakness is going to learn different types of movements and different types of skills and different ways to navigate the world than a child who doesn't have those issues to contend with. There's also plasticity, which isn't as much the idea of learning a skill, but just the fact that the brain will adjust to things that are happening. If your brain is not sending enough signals down to your spinal cord will turn up the volume and listen more intently to everything coming into it. That can be a good thing and partially compensate for signals, for the decreased input of signals to the spinal cord. But it can, when it goes too far, become a problem and lead to problems like spasticity, where you have excessive sensitivity to incoming signals. And the same is true within the brain itself. The injury to the basal ganglia that causes dystonia can, over time, lead to changes in other areas of the brain that are listening to that. Even to the point where if you fix the dystonia, the changes, where you fix the underlying problem, the changes in the other areas of the brain may not be fixed right away. This is one of the most interesting things about early treatments with deep brain stimulation, particularly in children with genetic forms of dystonia. And it was found that when you would either use deep brain stimulation or in some cases make a surgical lesion in the areas of the brain, children would improve, but it could take 6 months to a year for that to happen. And if you think about that, you've made a hole in the brain on day one, and they continue to improve 6 months out. That's not due to the hole in the brain directly. It's due to the secondary effects, and not just the immediate change in pattern, but a slow change in pattern that happens as the rest of the brain adapts to that hole. The same thing is true for deep brain stimulation. When we start stimulating, we often see effects within minutes or hours, but the full effects of deep brain stimulation could take months, in some cases a year or two, to see those full effects. So that there is the immediate change in pattern that occurs, and then there's the long-term change in pattern that occurs because of the plasticity. Which areas of brain have plasticity as a result depends on the pattern of stimulation that causes the plasticity, which changes the effect of the stimulation. And now the stimulation is now going into a brain that has itself changed because of these different patterns. And those new changed areas of brain will now respond in very different ways to the new types of patterns, and so on and so on. So there's this continuous change, and we find that after we put in the deep brain stimulation leads in children, you often have to keep changing the voltage or the frequency, and in some cases the position of stimulation, as the child's brain changes and adjusts to the pattern of the deep brain stimulation itself. So it's a very complicated situation, which involves the interaction between the disorder and the abnormal patterns it creates, the deep brain stimulation and the changes in pattern it can create, the plasticity response of the brain to the disorder and to the deep brain stimulation and eventually to their combination, and the learning effects as the child during development learns skills in order to be able to accomplish what they want to do in the context of the disease and eventually the deep brain stimulation. So the eventual goal of all of this is to understand this. Right now, when we put stimulation leads in, we make our best guess and we put them in. And one way in which we've been able to improve that guessing a little bit is by putting temporary leads in multiple possible targets and testing them. But of course, we can only test for a short time. We test the temporary leads over a period of about a week. So we can't see the plasticity and learning effects in that period of time. But we can at least figure out if we're getting some improvement in function. We can also measure to some extent the changes in patterns that are happening, but we don't know this at the level of detail and the ways in which these signals propagate through the brain. So the hope is eventually we will move from our current understanding, which is very much trial and error and a rough idea of what these things do, to a much more in-depth understanding where we can learn about how does disease affect the patterns of neural activity in the brain? How does it change? How does the disease function as a modulator that changes the information flow within the brain? It doesn't directly change the information, but it changes the information flow, and that eventually will indirectly change the information. And once we understand that, can we then predict the effect of different types of deep brain stimulation and different types of chemicals, and the ways in which those interventions will change that pattern into a different pattern, not necessarily a normal pattern, but a different pattern that will have a different effect on muscle activity. And when we understand all that, we will finally reach an age of neuromodulation, where we can take a brain that has been injured in some way, that is not functioning the way it is supposed to do, and we convert the pattern of activity of that brain into a more functional brain. And if we can do that, we can restore function to the child. We may never be able to reverse the underlying injury. We may never be able to fix the genetic problem. We may never be able to fix the brain injury or the stroke or whatever it was that happened. But we may be able to block the effect of that injury, block its expression, so that we don't see the effect of that injury, and the child does not feel the effect of that injury, and the child eventually will be able to function much better than they currently can. It's that level of understanding that we want to get to. We're not there yet, but we are at the early phases of this, and the technologies that are needed to obtain that do appear to be within sight. Thank you very much.